So I think it, maybe it goes without saying, but I think I should say it, that the work that David talked about, that I'm going to talk about, and that Phil is doing actually involves a huge number of collaborators, many, many more than I can list on one slide. Includes, Dalton Conley, Olga Rostopchova is here, James Lee, many, many other people. So this is just repeating David's slide, basically, on candidate gene studies. Some of the reasons that for the replication problems are listed here. One issue, echoing what Sarah said, is that weak hypotheses, often in the social science literature. David was just talking about population stratification. Another issue, which is an issue in many literatures, not just this literature, but uncorrected multiple hypothesis testing and publication bias seems to be a problem here. What I want to focus on in my few minutes is this issue of low power. So if you look at the literature in social science genetics, until very recently, almost all of the existing work has used sample sizes in the range of about 50 to 3,000. So 50 to 100 is the sample size for a typical laboratory experiment, and 3,000 would be a kind of large survey empirical analysis. So you can ask what sort of effect sizes would justify using sample sizes in this range. And the answer is that for 80% power for 50 people, you need an R squared of 8% for the SNP. For 3,000, you need an R squared of a third of a percentage point. And there's reason to worry that kind of effect size may not exist. In particular, genome-wide association studies with cognitive performance and personality, both with sample sizes of about 20,000 people. Had there been a SNP of an effect size a third of a percent, at the genome-wide significance level, they would have had 99% power to detect such effects. And in fact, in both cases, nothing was found.
So is this power for a single alternative? I'm just trying to understand the nature.
This is power.
A vector hypothesis test.
No, that's right. This is just for a single SNP, a null of no effect versus an alternative of 0.3%.
Okay, but you could imagine also a vector, SNPs that you're interested in, asking what any of them have, and you get a somewhat different power.
Absolutely, yes.
Right. Have you? I mean, just
polygenic scores.
Yeah. So this is not that. So one can ask, or what sort of effect sizes might be reasonable to expect. And it's hard to know because there isn't much evidence yet for, I think, for well-established associations with social science traits. But one place to look for what might be plausible is what are well-established associations in the medical literature that for traits that are most similar to the kinds of traits that as social scientists we may be interested in. So here are some credible estimates that have been replicated in large samples for smoking and the acetylcholine, one of the acetylcholine receptor genes, a SNP in that gene. The R squared is 0.5%. That's the strongest association with smoking that's well established. For BMI and FTO, which was mentioned before, the association is 1/3% R squared. For height now, it's an interesting case because now there's been GWAS, the latest GWAS is with over 200,000 individuals. There have been 150 loci associated with height. And among those, the largest has an R squared of 0.4%. Now, it's conceivable that the strength of the associations with IQ or educational attainment might be different from this. But I would expect that given that those, I think those traits are further from the underlying biology, I would expect that if anything, those effect sizes are likely to be smaller. So if that's true, then it seems that most of the existing work would be underpowered, which implies a high false discovery rate. So let me explain that claim. I think it's helpful to do a numerical calibration. So we'll take a simple case. So of course, genotypes are typically 0, 1, or 2 variables, the number of minor alleles. But to simplify, imagine there's just 2, high versus low, and imagine an outcome which is normally distributed. And to, again, simplify, imagine there's only two states of the world. Either there is no association or there's a true association. And if there is an association, its effect size is 0.02%. The reason I pick that number is because, as you'll hear from Phil's presentation, I think the best evidence we have so far for an association, a direct association between a SNP and a social science trait, educational attainment, is that magnitude. So what would be the sample size that you need for 80% power? It's about 40,000. So an order of magnitude larger than what had been standard in the social science work. Now, you can ask a different question, more of a Bayesian question, which is, suppose someone comes to you with a significant result. It's significant at the 5% level. What's the likelihood that that is a true association? So we can do a kind of heuristic Bayesian calculation given this very simple set of assumptions. And that's what's shown on this slide. So there are, of course, we need to assume a prior probability. I think 10% corresponds to a pretty optimistic prior, given, if we're talking about, say, cognitive ability, there are, something that's been estimated that 70% of genes have some kind of gene expression in the brain. So to put prior probability of 10% for any particular SNP is probably pretty optimistic. I think for a GWAS, something on the order of 0.1 to 1% may be a more appropriate prior. And the columns here show different sample sizes, which correspond to different amounts of power. Let's think about this case.
And can I just, I'm sorry, I'm a little fuzzy on the way to think about the prior.
OK.
So I look at one SNP in isolation, and my question is, was my a priori belief that it has any effect whatsoever, no matter how?
No, so this is for the simple case of either a null, there's only two states of the world, either null or true association of 0.02%, 0.02%. Yes. So if you start with a prior of 10% and you observe a significant effect in a sample of 100, your posterior should only go up, should barely move at all, go up to 10.4%. So, to a lot of us, that's counterintuitive. After all, a statistically significant result isn't that strong evidence. And the answer is that in this world of very small effects, something being significant at the 5% level with this kind of sample size is actually not that strong evidence. So a way to think about it is in terms of the power compared to the significance level. So the significance level of 5% means that under the null, there's a 5% chance you would have found the effect that you did. Power being just a little bit more than 5% means that under the alternative, there is a little bit more than 5% chance you would have observed what you did. So having observed what you did, that's like a little bit more than 0.05 to 0.05 ratio kind of strength of evidence, which is actually not very strong evidence.
But this calculation doesn't take into account any other studies that have been done on the same candidate gene.
That's right. This is for a single study.
For a single study, but in principle, there are a bunch of other studies. So if anybody does this incrementally.
But this is the standard methodology before these guys wrote this paper. I mean, I think this was meant more as a critique of the way everybody else was doing it, which is
right.
exactly
You can offer guidance on trying to accumulate the evidence across the country.
Oh, absolutely, yeah. So maybe if there had been 1,000 studies, or sorry, 100 studies of 100 people, then we should be looking at this column. If they all found the effect, we should be thinking about that as a sample size of, well, I guess we have to think about what the right significance level. Yeah.
Right.
I take your point that you want to.
There are independent studies. You don't have to pool them, but I just
Yeah. I agree. So you need to think about the right way to pool if there are multiple studies.
But here's the current practice is to look at each study as if it started from scratch, and there's never. So the significance level is, gee, today I'm the first one to ever look at this candidate gene.
Well, I think the current practice is to do a candidate gene study. If you find an effect that's significant at the 5% level, you declare victory and you say, yes, this is a positive result. It should really increase our confidence that this is an effect.
But they wouldn't do it like a meta analysis to go with it on all previous.
Oh,
studies.
yeah, there are
Don't
meta-analyses
forget
as well.
There are tons of unpublished, I mean, most of the stuff isn't being published because most of the time, people are getting no effect.
Right.
So, I mean, empirically, that's hard to do because if there are 1,000 people who've done it and only 2 published, you don't know what the end is.
So you could at least present a calculation of how many studies have to have gone unpublished to have produced something significant. If it's a small number, then it's not very, then you pass it out, if it's like 1 million, so I don't know. That's the standard way if people try to cope with it. It's not a clean answer, it's a way to think about it.
Yeah, I agree. Yeah.
Not that, you know, at NIH we have like what the sole arbiters of truth, but I can tell you that in peer review, what would happen to you if you did a GWAS study without, you know, an elegant or relatively elegant power analysis, sort of like this one. Even if you're looking for specific candidates, they'd want to see something like this, and if your power was like 24%, they'd tell you to get lost. Unless, of course, you had a meta-analytic plan in place where you're going to say, I'm going to take this data set and this data set, and this data set, and you could, you know, come up with a good argument. Or if you had an alternative analysis that came up with a better looking power analysis. But to be brutally honest about it, we have tended to completely discount previous candidate gene analyses when you start going genome-wide. And that's simply because, well, kind of what was Dan presenting before, since like only like 1 or 2% of them have ever replicated, you know, when I look at genome-wide, your prior should be kind of down in that 1% or 2% range, at least for things you can measure hundreds of thousands of people. And that's where there is, I think, some dialogue and dispute, is that, you know, you can imagine there are maybe phenotypes that, you know, where there are much larger effects. They just may not be social phenotypes like income or educational attainment.
So another thing I want to point out is that even if we go over to this column with 100,000 people, where power is almost 100% to identify an effect that's there, it's you still, 5% significance is not very strong evidence. I mean, you still have there's still a third chance that the result is a false positive. And that's because you're just learning at most. Get 20 to 1 evidence in favor of the hypothesis at a 0.05 significance threshold. So one of the things this kind of calculation highlights is that when we're in this world of very, very small effects, we're going to need both large samples and stringent significance thresholds if we're going to be doing hypothesis testing. So this is again just repeating a slide that David showed before, that in a genome-wide association study, now we're testing 2.5 million SNPs at a time in a typical gene chip, and we do have a much more stringent significance threshold, the so-called genome-wide significance threshold. That's an attempt to correct for the multiple hypothesis testing. David emphasized one advantage of GWAS, which is dealing with population stratification, but another advantage is that the same kind of power calculation or Bayesian calculation suggests that if you find something that's significant at the genome-wide significance level, then you can be pretty confident that it's true. So this is the same kind of calculation as before, but now with genome-wide significance as the significance threshold. And now if you look at a sample of 100,000, power is only 16%, so you're going to miss a lot, even with that kind of sample size. But if you find something, you can be quite confident that it's a true effect. But you still need these samples of 100,000 people or so. So that leads to another power problem, which is that there are relatively few data sets that have both the whole genome data and the social science outcomes of interest. So for example, for cognitive ability, the largest GWAS has had about 18,000 people. That's, it's because getting a high quality measure of cognitive ability requires administering a battery of tests. It's time consuming. It's not the main focus of the medical geneticists, who are the ones who are primarily collecting the genome-wide data. And this, as I mentioned, this GWAS found no SNPs reaching genome-wide significance. And if you do so what can we do about that? Well, what the medical geneticists have done is to form consortia where the way it works is that individual data sets run GWASs, and then upload results to a central server. And then these set of results are meta-analyzed across the many data sets. And it'd be better, of course, to pool the data, but IRB restrictions prevent that. So this kind of uploading to a secure server and meta-analyzing is a way to deal with that. And then, then the genome-wide significance threshold is applied. And so what we decided to do to try to imitate, learn the lessons of the medical geneticists and do something similar for social science. So Phil David and I formed the Social Science Genetic Association Consortium in 2011 to try to do this. Dalton is one of our advisors. Olga's a postdoc with us now. And we're basically doing exactly this strategy. And one of the key questions that, of course, we face is, well, what kind of outcomes to study? And there's a basic trade-off between having a larger sample, where you're typically stuck with a pretty crude measure because you're stuck with what happens to have been measured in large samples among the medical data sets, who are the ones that have the data, versus focusing on the social science data sets, or the small set of data sets where there's high quality measures but a smaller sample. So I want to make one last point, which is to think about this trade-off in terms of statistical power. These are ISO power curves. On the x-axis here are sample sizes ranging from 0 to 100,000. And on the y-axis are effect sizes ranging from 0 to 1% of the variance. And the different colored lines correspond to outcome variables that are of differential quality in terms of measured in terms of retest reliability. So the green line is a perfectly measured variable. You measure the outcome on 2 occasions, you get exactly the same answer. The yellow is a retest reliability of 0.2, so only a 0.2 correlation between one occasion and a measurement occasion and the next. So just to give a sense of the kind of conclusion that comes out of these sorts of calculations, think about an effect size of 1/10 of a percentage point, which I think is quite large for what we can expect for a single SNP for a social science outcome. If you had a perfectly measured variable, so let's say a measure of IQ from many, many tests, you would need a sample of 35,000 people to have 50% power. On the other hand, if you have a crappy measure, maybe just based on much shorter tests with only a retest reliability of 0.4, you'd need about 80,000 people for 50% power. In a lot of cases, it's going to be much easier to convince medical data sets to add a few questions for a crappy measure and get up to a sample size of 80,000 than it's going to be to get 35,000 people to take hours worth of cognitive tests. And a similar kind of logic goes for many kinds of outcomes that you might think about, like risk aversion, how you might go about studying that. It also relates to the issue that has come up before about the fact that in GWAS you're pooling across many different samples and thereby degrading the quality of the measures. You're getting different measures in different samples. A similar kind of logic may suggest that that's, if by getting a larger sample you can do that, it may be worth doing. So that's the logic that led us to have our initial kind of proof of concept project on educational attainment, which is in many ways a really crappy outcome variable, very far removed from the underlying biology. Means different things in different countries, and yet we decided to start with that because it's available in very large samples. Basically, every medical data set had measured it as an outcome variable as a matter of a demographic.